My name is Shai Carmi. I'm from the Hebrew University of Jerusalem, from the Faculty of Medicine. I work in population genetics and medical genetics, and this is one small project in quantitative genetics that I would like to present today. So the subject is screening human embryos for polygenic traits, which I think is quite interesting, as you will see. And so to get started, let's first define the problem, or define what we are talking about. So what is embryo screening, and why do we do it? So the idea of screening embryos, selecting embryos, is to generate embryos by in vitro fertilization, or IVF, and then genotype the embryos, test them for mutations or copy number variants, and then only use the embryos that are free of certain mutations. So this is called pre-implantation genetic diagnosis, or PGD. And when do we do it? What are the indications? So the main indication is when the parents are carriers for recessive mutations for Mendelian diseases like cystic fibrosis or Tay-Sachs in some populations, for example. And then, unless we are going to do this procedure, the parents are going to have probability of 1 in 4 to have an affected child. So we screen the embryos in order to avoid children who are homozygous carriers. There are some other indications like recurrent pregnancy losses, which are usually due to translocations in one of the parents, which leads to copy number variants or aneuploidies in the children. Sometimes people do PGD for sex selection when the disease is X-linked, so to avoid male children. And recently it also became common to screen embryos for diseases, for mutations in genes such as BRCA1 and BRCA2 that are autosomal dominant, meaning they can increase the risk of disease also in carriers. So recently this also became popular. So how does it work? So after IVF, the embryos are grown in a dish for a few days, usually 3 to 5 days, and then a single cell is extracted, as you can see here. Sometimes a few more cells if the embryos if we are doing it in 5 days, and then DNA is amplified, and then we test it, and then only embryos that are unaffected are implanted, hopefully leading eventually to pregnancy. This is not new. This has been around for over 20 years, almost 30 years now. But what happened recently over the past 5 years or so is recent technological progress that make these tests very accurate and a lot more widely used. So what happened is that obviously we have new sequencing and genotyping technologies, and the technologies for whole genome amplification also improved a lot, and also the statistical methods. And what we can achieve by now is we can get the entire genome of each embryo. We can get the haplotypes and copy number variants, even translocation sometimes. With recent methods, we don't need to tailor the procedure for each family. So it's kind of like a universal test. We don't need additional relatives like grandparents or uncles and so on. These tests are fast, accurate, and with the method that I presented here last year, we can do it for a relatively low cost of just under $100 per embryo. So how is it possible to achieve all of these? The intuition behind it is that if you think about it, the embryos are essentially just mosaics of the parents. So the parents are adults. We can genotype them from blood or saliva, and to high accuracy. And then to genotype the embryos, all we need to do is just determine where crossovers are. So as you can see here, obviously this will not cover de novo mutations, but otherwise we can get really good coverage of the genomes of the embryos, even when we're genotyping them on a sparse array or low coverage sequencing. So just in one slide, what I showed last year is a method that was since published. Actually, this is the same student who also worked with Juliana. She talked about his work yesterday. Quite interesting. Right? He wrote 2 papers in with PIs in 2 different continents the same year. So the way we implemented it is using a hidden Markov model, where the hidden state for each embryo is a pair of states indicating which of the two chromosomes of each parent were transmitted. And then transitions between states are due to crossovers, combinations, and to model low covariance sequencing, the emission probabilities take into account the number of reads covering each SNP. So again, this is what I showed last year. I don't want to go into more detail. This is just to show you that this is possible. We can get very accurate methods for PGD that are also relatively affordable and easy to use in the clinic. So in parallel, as you have heard in this conference, you saw this figure already, then the past 10 years or so witnessed a very fast growth of the number of successful GWASs, and by now we have thousands or even more variants that are associated with all kinds of complex traits. So based on these GWASs, what we can do is not only find these associations, we can also use them for prediction. Again, this was mentioned. I'll just summarize. So the standard way to predict traits or disease risk for new individuals that were not in the original GWAS is based on polygenic scores, and this is the key formula. Where in this formula, M is the number of SNPs that we include in the score. GI is the number of effect alleles carried by the individual. It can be 0, 1, or 2, and beta hat is the estimated effect size of each SNP. So the common practice in the field is only to include not to include all the SNPs on the array or all the imputed SNPs, but just to include those with strong evidence of association based on a p value that is sufficiently low, and also to remove SNPs that are linked. And then we can optimize these thresholds using cross-validation. So there are more sophisticated polygenic scores, but this is the basic method, and it actually works quite well. And to give you kind of an overview of what we can achieve with polygenic scores, then I generated this table with some representative values for some traits. So it's actually quite difficult to estimate the readability, and for some traits there are estimates that have very wide range. I try to use estimates from papers of Peter Vischer and Naomi Ray, assuming they would be here. But anyway, there are some other estimates. So for example, for height, they recently claimed that whole genome sequencing explains 80% of the variance in the trait. So irritability must be at least 80%. So H squared is the irritability. This is the proportion of variance in the trait explained by genetics, at least by additive genetic component. H squared SNP, is the proportion of variance in the trait explained by SNPs that are found on the microarray. And as you can see, it's always less than the irritability because of rare variance and copy number variance and so on. And then we have the variance explained by the polygenic score, which is of course always less. So for height, this is the trait for which we have the most data, and by now we can achieve accuracy of 25%. And as Peter mentioned a few days ago, he predicted it would be around 35% over the next year. For BMI, performance is lower, and then we have the cognitive traits like education attainment and cognitive ability, where we can predict around 10% or 5% of the variance, and for physiological traits like cholesterol or blood pressure, we can predict around 4% of the trait. It's a little lower, but still highly significant. So what did we learn from these two, apparently distinct, fields? One conclusion is that, kind of like looking together at what I just said, is that now it is feasible to screen human embryos for complex traits, because now we can genotype embryos very easily, and now we can predict traits very easily. So why not do it, on human traits, on human embryos? And actually, there is already one company who is offering such a test. It's called genomic prediction. So I give them free advertising here. As you will see, not so maybe not so free, but yeah, because of what I'm going to say in the next few slides. So from their website, they are They claim that they will screen for all these diseases, and they also published a paper a few months ago demonstrating the method that they are using to genotype the embryo. So the first author here is like really the world leading expert in PGD. So I really trust them that they get accurate genotypes. But what is perhaps disturbing is that they also testing for intellectual ability, and the way they do it is so you may be wondering, I mean, can we predict intellectual ability? But what they're doing is basically they're computing polygenic score for cognitive ability, for IQ, essentially or intelligence, and then what they're saying is that we're going to report embryos that have very low score for intelligence. So if this sounds disturbing to you, you're not alone. I mean, there are lots of ethical concerns. The key point is that it's basically eugenics, right? Because we're trying to control reproduction in a way to change the genetic composition of the population. So the first recent, at least, article to report it was in the MIT Tech Review almost 2 years ago, and then there was another wave of papers, designing babies and so on in this past winter. And then there were a number of geneticists and bioethicists that started writing about it about half a year ago. The New York Times panicked, like 4 months ago, and and so on, and more papers, more and more articles, and then obviously people say this is going to be really crazy, like in that movie, and so on. But what we need to have in mind is that this is just one view, which I assume most people hold this view, but some bioethicists actually hold the opposite view, that it's the obligation of the parents to provide their children with the best starting point, and hence it's also their obligation to to pick the genetically best embryo if they can. So there are some people holding this alternative view. Okay, so and then from the media now there are the discussions on social media and blogs and so on. The problem is so far that there was not much evidence. So one question is can this even work? So a lot of people who commented about this just say it's not going to work, but the company does say it's going to work. So so this is so there was not much reading about it. I mean, there were only a couple modeling analysis without empirical data. This is in social science, and they have some errors in this paper. It's just a blog. There's no paper with empirical data on what do we expect to see or what do we expect to gain from this from such a procedure. And this is what we did in our project. We used a number of alternative approaches to try to quantify what do we expect. What do we think are going to be the expected outcomes of screening embryos for polygenic traits? So the people who contributed to this project are the key people are Ehud is a master's student in my lab, and Otsuk, who was here a couple of weeks ago. Danny is finishing his postdoc here at UCLA, and Todd Lentz is a psychiatrist at New York. Okay, so let's first start with the first approach, which is using simulation. So how does it work? What is the pipeline? So first we start with real genomes of actual individuals, and then we pair individuals together either randomly or based on actual couples that we have in one of the data sets that I will talk about. And then once we have those parents, we will simulate, in a way that I will explain later, we'll simulate the genomes of a number of offsprings, let's say 10 children. And then once we have those simulated genomes of the children, we will compute the polygenic score for each child and then predict the trait. And then once we have the predicted trait for each child, we define the gain this way. The gain is the predicted trait of the top scoring child, okay? Minus the average predicted trait. So let's say we have 10 embryos. We rank the embryos according to their predicted traits. Okay, so let's say we are predicting height. And let's say the, the tallest predicted height is 1.9 meters, and the average predicted height in the family is 1.8 meters, so the gain will be 10 centimeters. This is the metric that we came up with to quantify the outcome of the procedure, and then we wanted to learn more about the behavior of this gain. And we focused on two traits, height, because this is the trait for which we have the most data, kind of a good model for every other trait, and then IQ, because this is also something that people are most interested in, as I mentioned before. Okay, so which cohorts did we use? For studying height, we used 100 Ashkenazi Jewish couples from a study of longevity, of lifespan. And for cognitive ability, we used around 1,000 Greek individuals, or just one second, this is a study of mental disorders. These individuals were just recruited to the military at the time. Yeah.
You add noise to the prediction?
I will get to that. Yeah, you will see now. Okay, and how do we predict these traits? So for height, we use the most recent meta-analysis from Peter Vischer's group that was published last year on 700,000 individuals, and then we use cross-validation to optimize the score with respect to LD pruning, so removing variants that are correlated, and the p-value threshold. And then for cognitive ability, again, we use the most recent GWAS for cognitive ability. And then again, we use cross-validation to optimize for the performance of the score. And then the results, if we try to use the score to predict the traits of individuals in this cohort, it works pretty much as expected. For height, the variance explained is around 25%, pretty much as reported in the original GWAS. And for IQ, the variance explained it's much lower. As expected, it's around 4%, again, very similar to the values reported in the original paper that generated these scores. Okay, and then how do we simulate embryos? So we have the parents. So again, the parents can be real couples or just random pairs. And then to simulate a child, that's a bit on the method here. To simulate the child, we take the two chromosomes. For each chromosome, we take the two homologous chromosomes of each parent, and then we simulate crossovers. First, we draw the number of crossovers according to a Poisson distribution. Okay. With mean equal to the length of the chromosome in Morgan's, and then we place those crossovers randomly, and then we shuffle the chromosome to generate the two possible gametes. We pick one gamete randomly, so we do that for one parent, and then we do that for the other parent. So we have two gametes, and then we unite them to generate the genome of a child, and then we compute the polygenic score of the genome, and then we predict the trait based on the regression line that I showed in the previous slide. Okay, so this is how we simulate. So now we have the genomes of the embryo. So now we can actually get to do some experiments and see what the expected outcomes are. So we use 10 embryos, which is kind of at the upper bound of what we can get from this procedure. And then again, we define the trait as the gain is the predicted trait of the top scoring embryo minus the average. And we repeat it over 500 families. Okay, so what do we get? Now is the interesting part where we finally see some results. So okay, so this is the distribution of the gain over the 500 repeats, and to walk you through these plots, then what we see here is the distribution of values of the gain. Okay, on the left side is height, okay, for random couples. In the middle is height for actual couples, and on the right is the gain for IQ. And what you can see here is that the gain is around 2 to 4 centimeters for height, and for IQ it's also 2 to 4 points. So numerically they're the same, which is really confusing, but it's actually completely coincidental. So just imagine that we had measured height in inches. So of course it would be. Now the numbers would be different. What's important, the way to understand it is that like 3 centimeters are about 1/2 of standard deviation of height. Whereas 3 IQ points, by definition, they are 1/5 of one standard deviation of height. So So this is quite important already because just by now we know what to expect. We know that if we are going to do this procedure, I think most people will say this is quite small. It's not a substantial gain. We can, like the best case scenario, we can increase the height of the children compared to, the average child by around 3 centimeters and increase the height by around 3 IQ points. And another point that we can see from these graphs is that actually there's no big difference between real and random families, meaning the role of associative mating is relatively minor, at least with these cohorts. Okay, and so this is like the basic result, and now we extended them in several ways. So the first question that crossed the mind of everyone who hears about it, so what happens, okay, if you don't have 10 embryos, what happens if you have many more? Maybe we can gain a lot more this way. So we can simulate as many embryos as we want, and if we simulate more, then so this is the gain versus the number of embryos, the mean gain. And what we can see is that I guess the benefit of doing this is rather limited. I mean, we can go from 3 centimeters. If we have 5 times more embryos, we can go from 3 centimeters to around 5 or just under 5. And in IQ, we can get to around 5 IQ points. And this number of embryos is completely unrealistic. So this is just a thought experiment. Maybe more importantly, what happens is that if you go down to 5 embryos, which is more realistic, then the gain goes down to around 2.5 centimeters or IQ points. And actually, if we are in this regime where we have 5 embryos or less, the number here is really crucial. I mean, if we go from 5 to like 3 embryos, we're really the gain is going to be much smaller. And so given these results, I mean, we also felt that there is a need for some theory, for a quantitative genetic model that will explain the results, but also more importantly, it will allow us to answer the question, what happens if you're going to have 10,000 embryos? Maybe it's too much to simulate, but we want to have a certain model that will give us the prediction for this. But more importantly, we want to know what happens if we have better predictor, if we can explain more variance in the trait. So the model that we used is the following. So it goes like this. Let me go over this. A few slides of explaining the math here. So we assume that so PS1, PS2, PSN they denote the polygenic scores of the n embryos that we generate. That we presumably generate in IVF. And then we assume that they have a multivariate normal distribution with 0 mean and covariance matrix that looks like this. So let me go walk you through this matrix. So sigma z squared is the variance of the trait, okay? And RPS squared is the proportion of variance explained by the polygenic score. So this term multiplies all the elements in the matrix, and then on the diagonal we have ones. It makes sense because this is the variance of the polygenic score by definition, right? It's the variance of the trait times the proportion of the variance explained by the score, and then on the off-diagonal elements we have 1/2 for all the off-diagonal elements, and this is because of it can be proven, but intuitively it results from the well-known fact that was also mentioned, I think, in Peter Vischer's talk, is that the covariance between in genetic value between siblings is 1/2 the variance. So this is kind of a well-known fact in quantitative genetics. Okay, so we have these polygenic scores, and now remind you that we define the gain as the score of the best embryo minus the average score. So if we write it mathematically, then the gain is the maximum of those n polygenic scores, okay, minus the average of these n scores. Now, if we take the mean, okay, then by definition we center the polygenic scores so that they have 0 mean. So the second term is 0, and then what we need to compute is the expectation of the maximum of these n scores. So how do we do it? So there is a little trick that can be done here, and we can think of these scores as the sum of two sets of multivariate normal variables, where and this is possible because the covariance matrix can be written as a sum of two matrices, okay, which we call y and z. So what is y? Oh, you can see the z. Okay, that's funny. So above the right matrix is z. Okay, so yeah, it's kind of very random. Okay, it's exactly covering the z. Okay, okay, so you can probably convince yourself that the sum of these matrices give us the covariance matrix of the polygenic scores. But now y is diagonal, so these variables are independent. And for z, the covariance matrix is all ones, so it represents identical variables. So it's basically just one variable copied n times. So because when we take the mean, because it's the same variable for all the scores, for all the n children, then it does not affect the maximum. So eventually what we get is that the mean gain is the mean of the maximum of n independent random normals. And this is quite a nice result. And why is this so useful? Because now we can use lots and lots of results that were proven over the years about the distribution of the maximum of independent normal variables. So there's a lot of literature on that. And we can use something called extreme value theory or order statistics. And it turns out like the maximum of n variables, under some limits, it has a distribution called Gumbel distribution. And we know the mean of this distribution. And this is the result where, it's a complicated term. The details don't matter that much. As I will show later, it's using some constants and normal CDFs and PDFs and so on. So this is the key equation, and it matches the simulations relatively well. But actually, we can gain more insight if we use a very rough approximation for the maximum of the independent normals. So it can be shown that approximately the mean of the maximum of n independent normals scales as the square root of log n. So we can actually write the mean gain in embryo selection like this, and it has two terms. So the first term is the standard deviation explained by the score. Okay, it's the standard deviation of the trait multiplied by RPS. And this is the contribution from increasing the number of embryos. So the fit is not less good, but it definitely captures the trend of what's going on. But now let's focus on the first term in this equation, which is sigma z, the standard deviation of the trait, times r, or maybe we better write it as sigma z times the square root of r squared, which is the proportion of variance explained. So as you can see, this fits the simulation pretty well. So the way we did it was, we cannot, unless we simulate the trait from scratch, but for height we cannot develop a better predictor, right? Because we only have the best predictor that we have. So we actually used worse predictors by using only subsets of the chromosome. So we kind of like diluted the signal to get predictors of lower accuracy, and then we can see that indeed the gain increases as the square root of r squared, meaning the square root of the proportion of variance explained. Okay. And this allows us to, now that we have this equation, we can also say, okay, what do you expect that will happen if we have better predictors in the future? So let me give you a couple examples of what do we expect to happen. So Peter Vischer said that he predicted that next year there will be 35% of variance explained for height. So let's say we have 40%. Which is, it's not, we don't have it now, but it's definitely within reach. So the gain will be on average 4 centimeters for height. For IQ, it's more difficult to speculate. The current score explains 5%, but assuming that GWAS sizes will grow over the next few years, let's say we explain 3 times the current variance, so 15%. So now the gain actually becomes more substantial. It's around 6 IQ points. We can actually we can say something about what do we expect to happen when GWAS sizes are going to be when GWASs are going to increase in size. So there is a formula, it appears also in Bogdan's paper, for the expected accuracy of the polygenic score, given the number of the effective number of markers on the chip and the GWAS sample size. So we can use it to predict what will happen when the GWAS sample sizes are going to grow, to reach sizes of 10 million or 100 million people. So it always has this S shape. So kind of diminishing returns for very high sample sizes, at least as long as we are using the same chip, which limits the the variance that we can explain. And so with height we are almost at the maximum. Maybe you can get to around 4 centimeters, but with IQ we have more room for increasing the gain up to around 6-7 centimeters. 7 IQ points. Okay. But so maybe this sounds quite a lot, like 6 IQ points. But we have to remember, and this goes back to your question. I hope it will be answered in this slide. Is that there is a lot of variance left. So so the mean gain is what do we expect to it's just like the average. The average. It's the average polygenic score of the best embryo. But what is it telling us about the actual trait value? So what is it telling us about the actual height of that embryo when that embryo becomes an adult? So there are a number of factors that we should take into account to interpret these gains. So first, there is some variability in the gain, as I showed you before. It's not always 3 centimeters. It can vary just because of random sampling. But it's very important to remember that there are unaccounted for genetic factors because the polygenic scores do not explain all the variance, and there is the environment, okay, or the non-genetic factors, and overall they will contribute substantially to variability in the value of the trait of that child. So what we wanted to derive is a 95% prediction interval for the value of the trait for the embryo that we select. So it's actually quite difficult to say something more accurate than that, but and the reason is that we can predict the trait of the child by not only using the polygenic score, but also using the phenotypes of the parents, which we usually know. So the trait of the child will be centered around ZMP, which is the mid-parental value. So this is the average value of the trait across the two parents, and then we have the gain, the mean gain, and then we have some variability that, to be most conservative, let's assume we can explain the entire readability of the trait based on both the scores and the value and the phenotypes of the parents, and this will add this term here, 1.96 sigma z times square root of 1 minus h squared, and this should be compared to what we expect without selection. Okay, and this was also in Peter Vischer's talk. He mentioned that if we know the average of the two parents, then the variance explained by just that alone is h to the power 4 divided by 2. So this is what we get without selection. So let me just give you an example to show you what we expect. So let's say again we have 10 embryos, and we can explain 40% of the variance using the score. So without selection, on average, if I just take basically a random male in the population, a 95% prediction interval for the height of that random individual, assuming the average is 176, is this range approximately. Whereas with selection, what we achieve actually, and this is really important. I mean, these numbers are really what we need to know about this procedure. With the selection out of 10 embryos, the confidence interval, the prediction interval will be 1.74 to 1.87 meters. So what it means is that we really here eliminated children that are going to be particularly short, but we don't. But it does not. I mean, it's still pretty wide range. It does not guarantee that the child will be particularly tall. So this is something important to have in mind, and it's actually more. The effect is more pronounced for IQ. So again, let's go back to 15% variance explained. So without selection, so the standard deviation of IQ is 15 points by definition. This is how it's defined. So without selection, it's around between 70 and 130. And with selection, again, what we're eliminating is just children with particularly low intelligence, but it does not. Essentially, as you can see, this range is very wide and does not guarantee that the child will be a university professor or something. So surely not at UCLA. Yeah. Okay. And I mean, once we have these formulas, you can plug in different values of what we have for the number of embryos, for the variance explained, and we can see, what do we get. So most people who look at these numbers think that the gain is quite limited, but I do need to say that for some people, it might be worth it to do this procedure. Then we have to think about all the ethics, whether we want to let them do it or not. So before kind of going to talking more about limitations and wrapping up, I just want to show one other experiment that we did, which is I think quite interesting. So obviously we cannot do actual experiments because it's unethical and we'll have to wait 30 years. So until we get the final height, right? So we took advantage of large nuclear families, and the idea is that we instead of generating 20 embryos, we're going to look at families with 20 children. So these families came from a study of height in the ultra-Orthodox Ashkenazi Jewish population. This study was done here by Leonid, and he contributed the data. And so these families are huge. I mean, they have on average 10 children per family, and some families have up to 20 children each. So again, we can use the living children to kind of do the thought experiment to say what would have happened if we just selected one of them based on polygenic score. So we have height and genotype for those individuals, and we can predict height with about the same accuracy as we did before. And there are some figures which I have if you want to see later, but the key figure is this because it's really demonstrating what's going to happen if you're going to do selection for height. So let me walk you through this. So each column here is one family. Okay, there are 28 families, and the gray circles are the heights of the offspring. Okay, it's all shifted to male height, adjusted for age and so on. So the gray circles are the children. Okay, the parents are the blue triangles, and the one child with the highest polygenic score for height is indicated as a red square. Okay, so now you can see, there are many families, and now we can look at some families. So for some families, the child with use the mouse. So for example, for this family, okay, you see the mouse here? No, okay, so never mind. So for for example, for these two families, 10 and 11, actually the child with the best score for height actually ended up being the tallest by pretty large margin. So in these families, if these were embryos, not children, if these were like 15 embryos or something, and we had selected the embryo with the best score, okay, we would have, eventually it would have resulted in the tallest child possible, okay? But so this is for these two families and like family 22 over there and a number of other families. But for some other families, actually there's a lot of variability here. So for some other families, for example, look at number 2 or look at number 14 and 15 here. Actually, the child that had the best score, the tallest score, he actually ended up being shorter than the family average. And this is because of all the genetic factors that we did not model and the environment. So if we do some statistics, then let's say we downsample all the families to just 7 children. Actually, the top scoring child is going to end up being the tallest only in a third of the families. And it's going to be shorter than the average, so we're going to actually have negative gain in 1/6 of the family. So on average, it's going to be short 3 centimeters, or a little bit more than 1 inch shorter than the tortoise. So as you can see, it worked in some families, but it's definitely not guaranteed. There is a lot of variability. Now, the last few slides, just to kind of think about other considerations that we have that will practically limit the gain of this. So I want to go through some of these considerations. So first of all, the prediction accuracy, we know it will be, it is supposed to be lower in families, in particular for cognitive traits. And this was shown remarkably by something like 4 papers. All of them came out in the past 3 months or so. And the idea is that when we have the genotype of the child, the genotype of the child is correlated with the genotypes of the parents, and the genotypes of the parents are correlated with the environment that the parents induced to their children, right? Because let's say the parents have the genes for high intelligence, then it is likely that they will invest in the education of their children, and so on. So in unrelated individuals, when we predict intelligence, we're actually also predicting some of the environment that the parents provide for their children. But within family, all the children have the exact same environment. So prediction accuracy is actually much lower. So this is kind of evolving topic nowadays, but definitely it will limit significantly the gain that we expect to get from such procedures. So as John mentioned yesterday, the polygenic scores are usually not transferable across populations. So all of this really holds for European populations. And another important point is that the number of embryos, so I assume 10 embryos usually through this talk, but the number of embryos decreases with maternal age, and also the rate of aneuploidy also increases very fast. As you can see here, you probably know about this, at age 35, as you can see, the rate of aneuploidy increases from around 30% to nearly 100% at age 45. So this will reduce the number of embryos that we can work with to begin with, and also have in mind that not all the embryos that we implant, they will be born, so we will lose some embryos. So effectively, we don't expect really to have 10 embryos. This is very optimistic. Usually we will have, I think realistically, around just 3 or so. More considerations is another consideration is that genotyping the embryo or calculating the score is not going to be as accurate as when we have saliva or blood from an adult. I mean, this can be improved by just doing deep sequencing, but then it will make it prohibitively expensive. There is the point about assortative mating, so it's positive for most traits. We show that the effect is relatively minor here, but as polygenic scores improve, some of their variance they explain is just due to assortative mating. And again, this is something we don't have within family, so it will reduce the accuracy of prediction. Another important point is what happens if we select for multiple traits. So not IQ alone or height alone, but what happens if we select for both. So it can be shown based on the model that if we select for M traits, the gain per trait decreases by square root of M. So the gain, if we select for lots of things, eventually we gain very little for each individual trait. And maybe most importantly, there's the point about pleiotropy. So when we are selecting for IQ, who knows what else we are selecting for? So actually height is positively correlated with lots of desired traits, but IQ, for example, is correlated with autism and anorexia, and maybe other things that we are not even aware of. This is very well known phenomenon in animal breeding, where, for example, cows now they produce it's like 5 times more milk a year than they did 50 years ago, because of very intense selection for just a small number of traits like milk production. But it also came with many severe diseases and health issues that they have. And now there is like a movement to select for not only for milk production but also for health and so on. But it just opens a lot of complications that we may not be aware of. So to summarize, at least with the current current predictors and a little bit optimistic view of how many embryos we can actually get, the gain is around 2.3 centimeters or 2.3 IQ points. If you're going to have better predictors, the gain is going to improve relatively fast, but not so fast with the increasing number of embryos. So using technologies like pluripotent stem cells, just converting them to oocytes and generating, thousands of oocytes, probably not going to be helpful. But this is at this point science fiction. And there is a lot of uncertainty that needs to be taken into account. And if you want to read more, you can see the preprint on bioRxiv. Thank you.